I'm Luca Mirimin. I'm a lecturer in aquatic ecology in Galway Mayo Institute of Technology in Ireland. And today I'll be sharing our experience with using a microfluidic high-throughput quantitative PCR system adapted to the monitoring and surveillance of aquatic species. Microfluidics refers to technologies that enable the handling and manipulation of fluids at nano and picoliter scales. They're generally cost and time effective because of rapid heat transfer and reduced sample and reagent consumption. The small volume enables the integration of sample preparation, target arrangement, and amplification and detection in the same device. Microfluidics devices have been particularly attractive to point-of-care applications in medical science, in other words, the lab-on-a-chip concept. And in the context of environmental DNA, you surely have heard about digital PCR enabled by microfluidics, but in recent times, the application of high-throughput qPCR system has also started to emerge. And that's what I'm hoping to talk more about in this presentation. So there's a number of approaches and technologies that have been enabled by microfluidics and small volumes. Some are liquids going through different areas at different temperatures to enable PCR. Others, the liquids are stationary in a compartment and temperature change, like in most thermocyclers. Isothermal nucleic acid amplification has enabled the development of disposable devices that operates at a single temperature, and then obviously digital PCR. Some of these technologies have been implemented in a number of commercially available platforms, and here are just some examples of those instruments. But the one I want to talk about today is the Biomark HD by Fluidigm. This is a high-throughput microfluidic PCR system and is capable of gene expression, SNP genotyping, digital PCR, among other applications in the same system. It's cost-effective, the throughput is scalable, and the controllers that performs a lot of liquid handling and mixing of reagents reduces the hands-on time. These are the integrated fluidic controls, or IFCs, which is where the magic really happens. So here, typically, the samples are loaded on one side, assay reagents on the other side, and then through pressure, the fluids are, the sample and the assay fluids are mixed into small microfluidic chambers of nanoliter volumes equivalent to droplet in a droplet digital PCR. And they are all partitioned independently, so it's not a multiplex, it's a parallel qPCR reaction. And within the center part of the IFC, there are, in this case, 9,216 microfluidic reactions occurring at the same time. So each of these samples is queried against all 96 assays in parallel, so not in a multiplex. One of the most attractive aspects of this platform is that with this system, the assays are not pre-spotted on the area of the chip, but rather they can be changed in every run based on improvement of the panels. So there's a lot of flexibility. The system has different throughput formats, and it includes a 37K digital IFC. This is the typical workflow. It includes a pre-amplification step, then followed by loading on the IFCs, and then the output based on the chemistry used can be probe-based or intercalating dye, therefore enabling high-resolution melt analysis. Just to give you a bit of an idea of the throughput, what the scale of throughput from a single run. So if we take a 96 by 96 IFC, it's running one single run is comparable to running 96 well plates in a conventional qPCR instrument, or 24 384 well plates. So you can imagine the amount of reagents, including template eDNA, as well as pipetting and time compared to a single run in the system. To illustrate the application of this platform to a project I've been coordinating for the last couple of years, and we are in the final phase now. I will talk about this as a case study. So the goal of the project was to provide a rapid and cost-effective source of data from environmental plankton samples capable of monitoring spawning events in aid of sustainable, effective aquaculture and fisheries practices. So here we're not. The goal is not to detect the presence of the adult population, so eDNA in terms of extracellular or extraorganismal DNA, but more like looking at viable larvae present in plankton samples, because normally microscopy is the way to go with it, and it's very time consuming. So in order to address this, we had two objectives: one, to develop a panel of assays targeting bivalve larvae and crustacean larvae in zooplankton samples in a semi-quantitative fashion, and also then to screen time series of samples. However, the steps that we undertook here are mostly overlapping with that of eDNA development of targeted assays. So this is just an example of the core species, among others, that we were targeting. So there's a number of bivalves and crustacean decapods. So we have oysters, mussels, lobsters, crabs, and prawns. We selected existing markers, we developed and designed new markers, tested them in silico. We used two different chemistries, a range of mitochondrial, cytoplasmic, and nuclear markers, generated universal crustacean and bivalve qPCR assays for internal positive controls, then use other synthetic IPCs for various applications. And in the first phase, we started with 22 assays, and we could validate rapidly 11 out of 22 assays. And now we've taken what we've learned from that phase, and we are testing 70 more assays. For the next step of PCR optimization, sensitivity, we played with a number of reagents to address inhibition. And also we looked at limit of detection and limit of quantification. So for inhibition, we looked at humic acid and EDTA at different concentration from left to right, with and without bovine serum albumin in the pre-amplification step. And we can see that when concentrations are high of the inhibitor, then BSA is very good at maintaining amplification and preventing inhibition. But we can also see that as concentration drops, that inhibitory effect is no longer there, and most likely because of a dilution effect of conducting reactions in nanoliter volumes. As for limit of detection and quantification, limit of detection was comparable to that of conventional qPCR assays, although we did observe a lot of variation in efficiency, and that's what we're working on. One other thing I forgot to mention is that the inhibition control was also tested on 66 environmental plankton samples, and we could observe that effect of the addition of BSA as a valuable inhibitor controlling agent. As for specificity of the assays, we tested, we did cross-species testing using both genomic and synthetic DNA using G-blocks, and also created mock communities with oligopools. And here we see an example of two assays whereby a specific amplification was observed in the target organism, but not in other off-target organisms. Similar performance was observed in the target when mixed in a mock community, and then a serial dilution of the mock community was used also as a standard. The numbers here also refer to the high resolution melt peak. Some assays were appeared very reliable across the species tested in vitro, whereas others were less reliable, so we had to tweak or discard them. But ultimately, this enabled fast specificity testing of a number of assays, and the intercalating dye chemistry combined with high resolution melting is very attractive and promising. Then we applied this panel to the screening of 242 plankton samples from 14 locations over 2 years, all around the country. These were vertical plankton toes collected by non-expert stakeholders and shipped by post to GMIT, where we extracted in bulk following an adapted power soil protocol. Just to give you a snapshot of the results, because these are unpublished and this is still with the funding agency, but just to give an appreciation of a single run, this is only a portion of a run. But we can see that from a number of locations, we can generate time series and presence, absence, and intensity of different species. And what we're working on next is to convert those intensity into to identify threshold, essentially, for all larvae against potential background noise of environmental DNA from the other population, as well as relative biomass estimation. So that's what we're busy with, including further validating the pipeline with barcoding of amplicons and high-throughput sequencing, and further explore the relative biomass estimation of spawning events. We're also expanding the panel to address other organisms and environments, both freshwater and marine, to detection of rare or harmful or unwanted species, such as toxic algal blooms, invasive alien species, etc. So to conclude, microfluidics and eDNA in the environmental DNA context. So it has capability to enable rapid and cost-effective development of targeted eDNA assay panels. Integration will lead to reduced contamination risks. Imagine the amount of pipetting that is saved. Allow fast turnaround time of screening of environmental samples, and avoid fast depletion of valuable template eDNA extracts. It could be implemented as a QC step or validation step in metabarcoding pipelines, and eventually is likely to lead to environmental point-of-care devices. I want to thank the funding agencies and other collaborators and the funding sources to enable this research. Here are my contacts. Feel free to ask questions now, but also feel free to reach out by email at any stage if you have any questions about this system or if you want to know more about it. Thank you for listening.